Now we're going to be looking at core principles. Now this is quite a tough section and it's quite long, so the two of us are going to be taking you through it together. Now this module covers the following topics: biological molecules, cells, transport of substances in and out of cells, exchange of materials between organisms and their environment, enzymes, and digestion. The molecules that biologists are most concerned with are carbohydrates, lipids, or fats, proteins, and nucleic acids. DNA and RNA are their derivatives, such as ATP. We are made of molecules, and it is their behaviour that determines the chemical reactions, metabolism, that keeps us alive and allows us to receive and respond to various types of information. So it is really, really important that you get to grips with this stuff. It'll make everything else easier to cope with. In order to understand the properties and functions of these molecules, you need to remember a little about atomic structure, particularly for the elements hydrogen, carbon, oxygen, and nitrogen. You will also need to know about the bonds between atoms, ionic bonds, covalent bonds, and bonds between molecules, hydrogen bonds, hydrophobic interactions, and disulfide bridges. Chris?
The following biological molecules all contain carbon covalently bonded with other carbon atoms and with hydrogen and oxygen. We say that all life forms on Earth are carbon-based. We shall first be looking at carbohydrates, which are divided into three types: monosaccharides, disaccharides, and polysaccharides. Glucose is a monosaccharide, or single sugar unit, found in grapes. It can exist in linear form or in a ring form known as a Pyran ring. There are two forms of glucose: alpha glucose, where the hydroxyl group on carbon 1 is below the plane of the ring, and beta glucose, where the hydroxyl group on carbon 1 is above the plane of the ring. Starch and glycogen are polymers of alpha glucose, and cellulose is a polymer of beta glucose. Fructose is a monosaccharide found in honey and apples. It has the same molecular formula as glucose, C6H12O6, but has a different structural formula. The ring structure here is a furan ring. Galactose is a monosaccharide in milk, also with the same molecular formula as glucose, but a slightly different structural formula. These three sugars all have 6 carbon atoms in their molecules and are called hexose sugars. Some important sugars in biology are 5-carbon or pentose sugars. Deoxyribose sugar in DNA and ribose sugar in RNA are both pentoses. You will learn more about these in Module 2. Notice that each carbon atom always has 4 bonds, shares 4 pairs of electrons, and has a valency of 4. Each hydrogen atom always shares 1 pair of electrons, or has 1 bond, and a valency of 1. And each oxygen atom always shares 2 pairs of electrons, therefore has 2 bonds and a valency of 2. Some sugars are disaccharide sugars. They are made from 2 monosaccharides condensed together. Maltose, found in germinating seeds, is made from 2 molecules of alpha glucose. Notice that a molecule of water is eliminated so that bonds can form between the monosaccharide molecules. The bond is called a 1,4 glycosidic bond, as it is between carbon 1 of one molecule and carbon 4 of the other. Sucrose, found in cane sugar, is made from 1 molecule of glucose and 1 of fructose. Lactose is found in milk sugar and is made from 1 molecule of glucose and 1 of galactose. Notice that in each case, a molecule of water is eliminated so that bonds can form between the monosaccharide molecules. During digestion, enzymes hydrolyze these disaccharides back to their monosaccharides. Remember that when we draw pictures of these molecules, the pictures are in 2 dimensions, but molecules have 3 dimensions. It is helpful to build models showing the molecules to see how the atoms are arranged. We can carry out tests to identify these molecules. All except sucrose are reducing sugars because they donate electrons to, a reagent called Benedict's solution. This changes the blue copper-2 sulfate to copper-1 oxide when heated to give a colour change from blue to red. Add Benedict's reagent and heat in a water bath. Sugars that have a free carbonyl or a free aldehyde group can react with Benedict's reagent in this way, giving a colour change from blue to red. They are therefore called reducing sugars. Sucrose does not react positively with Benedict's reagent, and although it is a sugar, it is a non-reducing sugar. It can be identified in the following way. If the Benedict's remains blue after being heated with sucrose, then the sucrose needs to be hydrolyzed. This is done by heating it with dilute hydrochloric acid in a water bath for 5 minutes. The test tube is then cooled, the contents neutralized and made alkaline by adding sodium hydrogen carbonate solution. Now the Benedict's test is carried out again and we see that it is positive. This is because the sucrose has been hydrolyzed to glucose and fructose, both of which are reducing sugars.
Polymers are large molecules, macromolecules, made from repeating units called monomers. Polysaccharides are polymers of monosaccharides. There are many polysaccharides, but you need to know about 3. Starch, which is found in plant cells and is an energy store. Glycogen, found in animal cells such as liver and muscle and is also an energy store. And cellulose, which can be found in plant cell walls and is structural. Starch is a mixture of amylose and amylopectin. Amylose is made from lots of glucose molecules, joined by 1,4 glycosidic bonds to give a long chain that coils like a spring. Amylopectin is also made from lots of molecules of glucose, joined by 1,4 glycosidic links to form shorter chains. There are also joins between carbon-1 of some molecules and carbon-6 of others to give branches to the chains. The presence of starch in plant cells and food derived from plants can be demonstrated by adding iodine to potassium iodide solution. If starch is present, the iodine solution changes from yellow-brown to blue-black. Heat is not needed for this test. Starch is present as starch grains in the chloroplasts of plant cells and in plastids. The plastids contain stored starch and appear violet when observed under the microscope. Glycogen is found in some animal cells. It is like amylopectin, made from many glucose molecules condensed together with 1,4 links and with 1,6 links forming branches. The whole molecule then rolls up into a ball with little bits sticking out. Enzymes can hydrolyze bonds here and snip off bits, changing them into glucose when needed for respiration by the cell. Cellulose is a polymer made from many glucose units condensed together. Every other beta-glucose molecule has to rotate through 180 degrees so that a bond can form between carbon-1 of one molecule and carbon-6 of another. Long chains of glucose molecules lie parallel to each other, and hydrogen bonds form between hydroxyl groups and oxygen atoms. Many hydrogen bonds give the molecule great strength. Lipids. That's fats to you and me. They are a diverse group of chemicals, the most common being triglycerides, usually known as fats and oils. Fats are solid at room temperature, for example, butter. But oils are liquid at room temperature, for example, olive oil.
Triglycerides are formed when one molecule of glycerol condenses with 3 fatty acid molecules. Fatty acids have a carboxylic acid group attached to a hydrocarbon tail. Glycerol is a type of alcohol. The reaction is a condensation reaction between each hydroxyl group of the glycerol and the carboxylic acid group of each fatty acid. This forms a triglyceride, and the bonds between the glycerol and fatty acids are ester linkages. The hydrocarbon tails are nonpolar and insoluble in water. Some fatty acids are saturated, with no double bonds between the carbon atoms. Others are unsaturated, having double bonds between some carbon atoms. The double bonds make these fats melt more easily. Oils tend to be unsaturated. Unsaturated fats should be eaten as part of a healthy, balanced diet, whereas the amount of saturated fat should be limited. Fats are insoluble in water, as they are non-polar molecules. They will dissolve in organic solvents, such as ethanol. Add some ethanol to a food that you think contains fat and shake well. Then pour the ethanol into another test tube that has water in it. Any fat that was dissolved in the ethanol now comes out of the solution in the water to form a milky emulsion. What are the roles of triglycerides? Well, they are energy reserves, as they have many carbon-hydrogen high-energy bonds. Fat, made of triglycerides, is stored under the skin as a source of energy, and when metabolized, it also produces water. This is known as metabolic water, particularly important in animals that live in dry areas. It also insulates the animal against heat loss, so animals such as seals and whales have a thick layer of fat, blubber, and humans, who are not hairy, also have subcutaneous fat. Phospholipids are formed by the condensation reactions between one molecule of glycerol, two fatty acids, and one phosphate. The phosphate group is polar, and so is hydrophilic and will dissolve in water. The rest of the molecule, the fatty acid tail, is hydrophobic. At a watery interface, phospholipids will line up so that the hydrophilic heads are in the water and the tails are out of the water. At the edges of cells, a phospholipid bilayer forms, with the hydrophilic heads of each layer facing outwards in the watery cytoplasm or in the watery extracellular fluid, and the hydrophobic fatty acid tails facing inwards. This forms the basis of the cell membrane. Some fats, such as cholesterol, have a ring structure. Cholesterol strengthens animal cell membranes, and from it we can make hormones such as testosterone and estrogens. A type of cholesterol is stored under the skin. It helps waterproof the skin, and ultraviolet light acts on it to convert it to vitamin D. Cholesterol is particularly important in nerve and brain cells. Proteins are macromolecules. They are polymers of amino acids. Each amino acid has a backbone consisting of a carbon atom, an amine group, and a carboxylic acid group. There are 20 amino acids involved in protein synthesis, and therefore important to biology. Each has a different side chain or R group. The simplest amino acid is glycine, with an R group being just one atom of hydrogen. Amino acids that have side chains containing oxygen or nitrogen have polarity and are hydrophilic. Those with just hydrogen and carbon in their side chains are hydrophobic. Remember that all proteins contain nitrogen, and some contain sulfur in their side groups, because some amino acids contain sulfur. Peptide bonds join amino acids together. The hydroxyl group is lost from the carboxyl group of one amino acid, along with a hydrogen atom from the amine group of another amino acid. This forms a molecule of water. A peptide bond forms between the carbon atom of the carboxyl group and the nitrogen atom of the amine group. Two amino acids joined in this way are called a dipeptide. When many amino acids are joined by peptide bonds, the resulting macromolecule is a polypeptide. And if there are 100 or more amino acids present, this molecule can be called a protein. The sequence of amino acids in the protein is its primary structure. This sequence is determined by the genetic code in a gene, which is a length of DNA. This chain of amino acids then coils into an alpha helix or a beta pleated sheet. This is the protein's secondary structure. Hydrogen bonds hold the coil or sheet in its configuration. The secondary structure then coils or folds into a complex and precise three-dimensional shape, the tertiary structure. The specific shape of each protein enables it to carry out its function. Bonds between side chains of certain amino acids hold the 3D structure in place. So, if the primary structure is wrong, the tertiary structure will not be correct, and the protein will not function. The four types of bonds that help hold proteins in their shape are hydrogen bonds between polar groups. These bonds are broken by high temperatures and by pH changes. Disulfide bridges form between cysteine molecules. These are covalent bonds and so are very strong, but they can be broken by reducing agents. Ionic bonds between ionized amine and carboxylic acid groups. These ionic bonds form when an electron from one atom leaves its outer shell and moves to the outer shell of another atom. The atom that has gained the electron is reduced and now has a negative charge, so is a negative ion, and the atom that has lost an electron now has a positive charge. It is oxidized and is a positive ion. The weak attraction between these two ions forms an ionic bond. Ionic bonds are weaker than covalent bonds. Finally, hydrophobic interactions occur between non-polar side chains. Proteins that are made of more than one polypeptide chain have a quaternary structure. Hemoglobin consists of 4 polypeptide chains. It is a globular protein and nearly spherical. It is present in red blood cells and carries oxygen from lungs to respiring cells. Enzymes are also globular proteins. The amino acids with hydrophilic side chains are towards the outside of the molecule. Those with hydrophobic side chains are inwardly facing. So they are usually soluble, since water molecules can surround the hydrophilic side chains. Some proteins form long strands, and these are called fibrous proteins. These are usually insoluble and are structural. Examples are keratin in hair, collagen and elastin in skin, actin and myosin in muscle. The amine groups in the side chains of some of the amino acids of a protein or at the site of ionized peptide bonds react with copper ions to form a complex that has a violet colour. Biuret reagent consists of 2 chemicals added to the protein in turn. A dilute alkali, such as sodium or potassium hydroxide, is added, followed by dilute copper 2 sulfate solution, and the violet-purple colour indicates the presence of protein.
Chromatography. You have seen some of the biochemical tests that can be carried out to identify certain biological molecules. Chromatography is another technique that can be used in the chemical analysis of substances. It involves separating molecules so that each can be identified and investigated further. Paper chromatography can be used to separate mixtures such as sugars, amino acids, or the photosynthetic pigments. The mixture of photosynthetic pigments is spotted onto a special point called the origin. Each spot applied is dried and many spots are placed at the origin. The strip of chromatography paper, or the plate, if it is thin layer chromatography, is dipped into a special solvent so that the end of the paper or plate is in the solvent but the spot isn't. A stopper is put in place so that the chromatogram is in an enclosed atmosphere that becomes saturated with the solvent. The solvent moves up the paper or plate, and the molecules in the mixture each have different solubility in that mixture. The molecules are also of different sizes, and they each move at different speeds with the solvent. This means that some move further than others, and they separate. None of them moves further than the solvent. When the chromatogram has finished running, it is removed from the solvent and a pencil line is drawn to mark the solvent front. It is dried and the distance from the origin to the solvent front is measured, distance y. The distance from the origin to one of the pigments is measured, distance x. The value x over y is calculated. This is the RF value for that molecule. The RF value can be found for each pigment molecule. The RF value shows how far the molecule moves relative to the solvent.